Hi, my name's Cameron. I'm a PhD student working in Professor Juan Perry's lab, and I'm going to be talking today about some of the results that we've generated, which primarily involve the use of liposomes for the use within vaccines, but how we can then use these liposomes with existing technologies to help us achieve these goals in a scalable fashion. So it's probably a bit of an understatement at the moment to say that vaccines are a fairly hot topic at the moment, and as we can see, the number of publications since 1981 has been on the rise. Where nanoparticles have been used along with vaccines. And it was only until today, when I was recording this talk, where Moderna released some really promising figures for their corona virus vaccine, which further indicates the need or the advantages that lipid-based products have within this field. So the role of nanoparticle really comprises of two aspects. The first being is that we need our antigen at the site of action where we can deliver it, protected, and it's able to elicit its action. But also secondly, if our payloads such as mRNA or protein requires the cell to be activated, our formulation or nanoparticle can do that, such as if we're able to activate an antigen presenting cell. So this can be achieved, but it's not limited to two different methods, such as which include absorption or encapsulation. Both have benefits, but with absorption, if our product is negatively charged, such as mRNA, it allows us for an electrostatic interaction to take place if our nanoparticle is cationic, which allows us to give a very high payload loading. But if we encapsulate it, it does give us slightly more options in terms of what the formulation we can choose, as it doesn't rely on any electrostatic charges. But as a consequence of this, it does lead to lower loading. But regardless, we have plenty of options to choose from. But if we look at the absorption method first, it's clear that using nanoparticles clearly plays a strong role in the administration of self-amplifying RNA vaccines. From the results, the use of cation lipids was able to produce comparable antibody titers when compared against a benchmark ionizable lipid particle. From this, the use of cationic lipids within the vaccine field clearly plays a strong role and is notably less expensive with a well-investigated safety profile. From this, by taking known cationic lipids such as DOTAP and DDA that was used in these experiments, it's possible to greatly facilitate and accelerate the already rapid development of these vaccines during times when we need them most. Therefore, in order to keep up with the momentum that this field has been developing at, we need to really move away from the traditional lab-scale production methods using round-bottom flasks and downsizing methods such as the use of sonication, high-shear mixing, in order to create our homogeneous suspensions of sizes around about 100 nanometers, as from the reasons I've listed below, they're expensive, they're loss of drug loading, can also damage, and they're also incredibly batch variant, which for obvious reasons regulators look down upon, as they want to ensure that each product being released from quality control has consistent characteristics. Because of this, we've been experiencing a bit of a bottleneck in terms of the number of lipid nanomedicines that have been released into the clinic, as there's clearly been a great amount of research being conducted into their use. However, there's been a lack of translation into actual viable products that can be used. And so because of this, scalability should really be built in from the offset in terms of the manufacturing process, which is why within our group we've been focusing quite heavily on using laminar flow technology with a cartridge design and a machine that I'm sure everyone here is aware of. We also then use a scalable form of purification in the form of tangential flow filtration as a way to filter and purify, but also concentrate our products, but also in a scalable fashion. So just briefly, the mechanism behind this scalable solution can be summarized in the following slide. Within one let, we have our lipids, which are solubilized in a water-miscible solvent, and in the other, we have our aqueous phase containing the buffer we wish our formulation to be suspended in. In this inlet, we incorporate our active pharmaceutical ingredients, along as it's hydrophilic in nature, such as RNA or protein. If it was a small molecule, for instance, that was more lipophilic, this could be immersed within the lipid phase in the solvent inlet, which will be incorporated within the lipid bilayer. Once these phases are loaded in the system, they can be ran to produce our formulation. Within the chip itself, as the fluids are under laminar flow, chaotic advection can be used to facilitate the mixing, which can be achieved by introducing chevrons or obstacles to increase the surface area between the streams. In doing so, by mixing the two streams together, it leads to lipids to nanoprecipitate and self-assemble at the interface. And as they flow down the microchannels, the hydrocarbon chains within the lipids starts to bend in order to reduce the contact within the aqueous phase, which causes them to eventually form vesicles. The flow rate of the organic phase and aqueous phase can be increased, which will lead to a more rapid dilution of the alcohol concentration, and as such, will reduce the time needed for the vesicles to stabilise, as there will be less fluctuations in the alcohol concentrations. And this has subsequently been identified as a critical process parameter within the microfluidic manufacturing platform, but this will be discussed a bit later on. So as I just mentioned, the flow rate ratio, which is the ratio of the aqueous to organic phase, has been established as a critical process parameter, and is quite widely known within the microfluidic field. But in order to investigate if any other additional process parameters exist, we set out to identify if solvent selection can be used as an additional tool which can be utilised along with other process parameters to tailor vesicle sizes to desired ranges, which can be useful when designing a vaccine formulation, as it has been shown that by tailoring vesicles to a smaller size, that they can move away faster from the site of injection than larger particles. The selection of solvent within a microfluidic system must be water soluble, and as shown from the figure, the water miscibility is directly related to the carbon chain length, with the shorter carbon chains being completely miscible, as hydrogen bonds can form between the water molecules. However, in order to ensure patient safety, and it's at the centre of formulation development, solvents that are defined within the class 3 bracket, such as ethanol or isopropyl alcohol or IPA, should be favoured over class 2 solvents than methanol, and this is outlined within the European Medicines Agency and within the guidelines listed. As others have also reported, influencing the solvent polarity can have an impact on the formation of liposomes. Therefore, within this work, we set out to evaluate the impact of solvent selection by characterising liposomes using early screening studies all the way through to larger volumes. So in order to test this, a rapid screening study was conducted where we used a range of different liposome formulations with a combination of phosphatidylcholine and cholesterol, and one with a PEG coating at which was formulated at a 3:1 flow rate ratio. From these early studies, it was found that the soy PC and the POPC liposomes had the greatest impact from the effect of solvent polarity, where they're roughly double in size when manufactured in IPA rather than methanol. Other than the pegylated formulation, there was an increase in size as solvent polarity decreased. Other than the size, there was absolutely no effect on the polydispersity either. This was then tested using formulations that are either cationic or anionic in nature. As it can be seen, using the 3:1 flow rate ratio like we did with the neutrals, there was a general increase in size for both formulations as a result of reducing solvent polarity, and there was no effect on the zeta potential, which remained between minus 15 to minus 25 millivolts. In comparison to the other two slides, the cationic formulations were found to be more susceptible to the effect of solvent polarity, where we can see an incremental increase in the formulation size using DOP DOTAP of roughly 50 nanometers when we moved from methanol to ethanol to IPA. It was not possible to size the HSPC cholesterol DOTAP formulation IPA due to aggregation, but interestingly, this effect was negated once PEG was added, and this was also seen within the neutrals as well. In order to investigate this further, cholesterol and PEG content within the formulation was altered. By looking at figure A, the same trend was observed, whereby the lower polarity solvents led to reduced vesicle sizes, but increasing the cholesterol concentration across all three solvents led to reduced sizes, and above constant cholesterol concentration at 22% allowed for uniform vesicles. But anything below this, unless made in methanol, the PDI was not recorded, or any vesicle size was not recorded due to aggregation. Interestingly, the carbon effect that was previously seen with PEG was also observed, but above concentrations of 8%, whereby the polarity, the effect of polarity was not seen. So we can maybe begin to explain the result through suggesting that this effect could be due to the alteration of phospholipid packing densities, whereby the research previously conducted has shown that by using differential scanning calorimetry and including cholesterol concentrations at 33 or 50% no longer makes the transition temperatures detectable and avoids the gel to liquid phase transition through the improvement of the phospholipid packing densities. So similarly, the presence of PEG lipid concentrations above a certain level could alter the geometric packing of the phospholipids within the bilayer, which would later negate this effect, and so not experience the same size changes as observed. So seeing this effect very early on was quite fundamentally interesting. However, why is it so important that we can influence the size and the effect, and what is the importance of the effect of charge within a vaccine formulation? But from this figure here, we can see that charge can be used to really direct distribution of our product, as the previous research has shown that cationic formulations remain at the site of injection, while neutral and anionic particles distribute more rapidly away and can drain away to surrounding to the lymph nodes, which is can be shown in Figure A here, where we can see a DSPC cholesterol draining quite fastly. However, if we go to our cationic formulation, in their DSPC cholesterol and DOTAP, we're able to maintain and stick at the site of injection for far longer. And if we see at the lymph nodes here, that the anionics are DSPC cholesterol, PS, and the DSPC cholesterol neutrals can be found at the lymph nodes in far greater quantities than the cationics. And as we can see in this figure, we can really show the importance of size in terms of wanting to be able to and how we want to distribute our formulation. And as we can see, that larger formulations are able to be maintained at the site of injection for far longer than smaller vesicles, which we can see drain away far faster and are present in far greater quantities at the lymph nodes than larger formulations would be. So being able to have another process parameter that allows us to manipulate the size can really give us a bit more flexibility in being able to create a formulation and get it to be distributed to whichever location that we wish. So for example, based off of these findings, if we want to create a formulation but make it stick at the site of injection, we maybe want to think about creating it more cationic and also slightly larger. But if we want to drain faster towards the lymph nodes, we can maybe think about it having more of an anionic or neutral nature, but also being in a smaller size to facilitate this effect. So by delving deeper into the neutral formulations, we really started to look at how we can tailor specific sizes in a more reproducible fashion. And we could really see this effect when jumping between methanol to IPA, where we saw the size being almost double, roughly going from 45 to 93. But in between those steps, there was an incremental size increase. So now we can begin to start to establish almost size tuning our liposomes to desired sizes. But regardless of what solvent composition was being used, uniformity across all of our samples was maintained below 0.2, which is the value that we've been using to demonstrate good homogeneity. This effect wasn't just limited to neutrals, we also tested it with DOP-DOTAP, and again we can see the size tuning effect by changing the solvent polarity methanol and ethanol, and by actually adjusting this ratio, the size was increased from 33 nanometers to 60 nanometers, all while maintaining the PDI below 0.2. So this effect isn't just limited or constrained to one formulation, it can be applied to multiple ones and of different charges. So from this we can start to build the foundations and maybe start thinking about this as being another critical process parameter to start thinking about, in addition to ones such as our flow rate ratio, our lipid concentrations, and our total flow rate ratios. So in our neutral formulations, we went with an entrapment method as opposed to the absorption method as discussed earlier. And ovalbumin, the protein, was used as an antigen analog in order to identify if there's any change or any benefit in the API loading by encapsulating the protein within the liposomes. From figure 10a, we don't see any benefit at all or any significant difference in pairing the liposomes in either methanol or ethanol. However, protein loading was significantly reduced when formulating the liposomes in IPA when this was used within the manufacturing process. And after then looking at the morphology images from the cryo-TEM, we found that small unilamellar vesicles were observed in the methanol and the ethanol. However, in the IPA, liposomes were seen to be larger, but with multiple bilayers seen in the images, and this could be due to the larger lipid discs being formed as a result of the improved solubility of the lipids in the IPA, and as such leading to more time for the vesicles to close when during the manufacturing process, leading to these multiple bilayers. So in terms of stability and stability though, there wasn't any benefit at all or improved stability as a result of changing between the solvents when stored at fridge temperature 208 degrees. Similarly, there was no difference in the release profiles either, with approximately 80% of the protein being released after 72 hours, despite the morphological differences. So from these results, it's clear that solvent selection does have an impact on the liposome and lipid-based products when manufactured by microfluidics, and it can be used to reduce formulations with desired properties, such as having a small or a large vesicle in order to influence the movement of the particles within systemic circulation. So with these process parameters in mind, the next question would be how we can use these parameters in order to scale up a formulation. So there are obviously quite a lot of micromixers available to be used, and clearly from the table and the figure that I've just shown, there are plenty of options available. However, a lot of these do come with inherent disadvantages. For example, the hydrodynamic flow focusing leaves the formulation quite susceptible to fouling after a while, and with the staggered herringbone mixer, their use for higher volumes needed for GMP production is quite limited in the fact that the structures within the cartridge itself is quite difficult to achieve throughput speeds that meet good manufacturing practice in terms of the final product volume, and the structures within the cartridge itself is also quite complicated to fabricate on a larger scale. So therefore, in order to address this, an alternative design based on our toroidal micromixer was developed, and this offers the potential for allowing for higher throughput of the formulation by creating Pareto advection through increased the number of vortices and centrifugal forces between the columns of the cartridges, which allows for these higher speeds to be obtained. So in light of this, what we wanted to test was if we can bridge between the two cartridges and maybe take one formulation all the way from basic R&D in terms of the formulation of the product, and then scaling this up to preclinical volumes and then all the way through to GMP volumes, which almost paves the way all the way from researching your initial product and testing it and then actually manufacturing it without needing to change any of the process parameters. So using our 3 unloaded formulations, we found that there was no differences at all between the sizes being produced across either cartridge, and there was no effect on the charge either. We then moved across and looked at the effect of incorporating protein, then our anionic and neutral by entrapment, and using PolyA as an RNA surrogate, which was absorbed onto the DOP. So we found that with the anionics there was quite a bit of a large size difference in between the unloaded and loaded, but again no differences between the cartridges, and there's only a slight size increase between entrapment with the neutrals and absorption with the cationics as well. Typically, we do expect really high entrapment efficient or loading capacities with the PolyA, which was observed, and we also saw roughly between 20 to 35% entrapment using the anionics, and between the anionics and the neutrals, 20% for the anionics and roughly 35% for the neutrals. Again, there was no charge change or any differences in the loading capabilities between either cartridges. So this was also encouraging in terms that we were able to quite easily bridge between the two cartridges with no problem at all, knowing that we can achieve the same formulation characteristics that we've been getting using the old design. And to confirm what we saw previously in the slides, we then decided to test the lipids that are different chain lengths. So the DNPC has a chain length of roughly 14 carbons, whereas the SPC and HPC has 18. And this confirms previous results where we've seen shorter carbon chain lengths having larger vesicles due to less van der Waals forces occurring between the carbon chains, and the DHPC and the HPC producing roughly the same size vesicle sizes, and with again no real changes in the loading capabilities either. So from these results, we can quite clearly see that we can interchange between the two cartridge designs without any problems at all. So without delving too deeply into the process parameters that we investigated, we did conduct quite a detailed study looking into the flow rate ratio and the total flow rate for each system being investigated. All this is within the publication that's listed on the slides, but as you can see, between the two cartridge designs there's absolutely no impact at all on switching either cartridge in the staggered herringbone or the TRM or next gen design. Or any effect in loading, and the same can be said when we looked at the solvent as well. So again, we saw this slight change in the fact that changing methanol to ethanol, we did see a larger size increase, but by changing the lipid concentration, we didn't really see much of a size difference or any significant differences in terms of the loading percentages, which again was quite nice, as it shows we can work at a lower concentration, so a less expensive formulation, but we can still achieve the same sizes and the same loading capabilities that we could get with a higher concentration, a high concentrated sample. We also conducted a release study and looked at the lipid percentages to see if there was any lipid loss at all throughout the process, before microfluidics obviously be 100%, and after we were able to achieve total recovery. We also looked, as for the release study, as you can see, by not entrapping the ova within our formulation, this is very quickly released in roughly about 24 hours, but as soon as we entrap it within our formulation, you can see that it's slowly being released over the subsequent days, and this was expected in the fact that there would be no differences between the release rates in the cartridges, as shown from the cryo images, whereby the liposomes are really nice and uniform and the sizes are approximately similar as well. We then looked at investigating biodegradable polymers, and in this case we decided to use PLGA, particularly due to its favourable degradation and the possibilities for sustained drug delivery as well. And from the results, much as you would expect, there wasn't really much change at all jumping between the two cartridges. The sizes were able to be nice and well maintained, and as well as the PDI, showing that we're able to achieve quite a homogeneous dispersion just using PLGA. And again, as you can see from the intensity plots, pre and post dialysis, we're able to get nice uniformity without any change switching between the cartridges. And the same can be said once we make these formulations cationic as well, where we can show that we can make nice small vesicles cationic that are cationic in nature, and regardless of what cartridge size you use, they are both approximately 50 nanometers. So just to kind of wrap up this bit of research, as a part of my project, I was able to go to P&I and use their GMP system, which employs their next-gen technology, and I was able to directly track my formulation using the neutrals and the DSPC cholesterol from the research and development side of things, in terms of using the Ignite as an R&D tool, to then go to preclinical testing using the Blaze, all the way through to GMP, where I was able to use their GMP facility and manufacture my formulation at roughly about 200 mils per minute. And as you can see, regardless of what piece of equipment I used, I was able to maintain the size and the PDI, and also using the GMP system, ensure that I was able to meet the volumes or the protein loading that I've been expecting as well from my basic R&D research as well. So from this, I was able to fully translate my formulation from the start all the way through to the finish, which really demonstrated the scalability of the system and working within microfluidics, and a clear advantage in using this manufacturing platform. So therefore, to conclude, the results have hopefully demonstrated a fast, low volume and high throughput microfluidic manufacturing system that allows for early screening of lipids and active pharmaceuticals ingredients where quantities may be limited. We have validated a controlled process from early R&D all the way through to GMP, which allows for the development of medicines with the same manufacturing characteristics without needing to change any of the process parameters. And we've also seen that microfluidic manufacturing as a flexible production platform, which can use different microfluidic geometries to formulate our liposomes without any change to their characteristics. And finally, we have established an additional microfluidic process parameter, which can be used to tailor our formulation to meet regulatory requirements. And with that, I'd like to conclude my talk